Okay, so welcome back to this next video in which we are discussing the vascular endothelial growth factor receptor. Okay, so we've seen that if you have a cell which is hypoxic, it's going to start releasing vascular endothelial growth factor. Now we want to look at the different types of vascular endothelial growth factor. Specifically, we want to look at vascular endothelial growth factor A, because that's the main one that is going to be increased in expression in response to hypoxia. Okay, so, right then. First thing, let's just write out the name in full. So VEGF is short for vascular endothelial growth factor. And basically there are 9 different families of vascular endothelial growth factor. Sorry, not 9, 7. Don't know where I got 9 from. 7 different families of vascular endothelial growth factor. Okay, and these are vascular endothelial growth factor A, vascular endothelial growth factor B. Okay, so let me label some of these up. Vascular endothelial growth factor A, vascular endothelial growth factor B. Then there is also vascular endothelial growth factor C, and it goes on all the way down to F. So I'll write them out. We're all halfway there now. Vascular endothelial growth factor C, vascular endothelial growth factor D rather. Okay, vascular endothelial growth factor E, and then finally we have vascular endothelial growth factor F. Right. Now, there is actually a 7th one, because I said originally that there was 7. So the final one is something known as PLGF. Okay, so it's not called VEGF, but it is considered a member of the family of vascular endothelial growth factors. And PLGF stands for placental growth factor. Okay, so they are classified according to their structure, and placental growth factor has the structure of a vascular endothelial growth factor, basically. Okay, so it's put in the same family. Right, so we are going to concentrate completely on VEGF-A, okay, because this is the one that's going to be upregulated in response to hypoxia. Okay, so there is only one gene for vascular endothelial growth factor A. Well, there will be 2 genes because, of course, you have homologous chromosomes. You'll have 2 genes, one from your mother, one from your father. But there's only one gene, okay? On a haploid set of chromosomes, there will only be one gene for VEGF-A. Okay, now, however, there are 8 different isoforms of VEGF-A, okay? And these come from the alternative splicing of the primary transcript for VEGF-A. Okay, so let me discuss this. So basically, if we have the VEGF-A gene here, so let's have our DNA here, and this will be the VEGF-A gene. So I'll colour it in. So this boxed here is going to be the VEGF-A gene. Okay? Then what will happen is RNA polymerase II will come along. It will transcribe the coding strand of the VEGF-A gene, and it will produce a piece of mRNA which is complementary to the coding strand of the VEGF-A gene. Okay? And this initial piece of mRNA it produces is what's known as the primary transcript. Okay, so this is the primary transcript, or the pre-mRNA, so it's prior to it being spliced. Now, this is going to contain exons and introns, and the exons are the pieces which will actually be used for translation, basically, and there are going to be 8 of these. So I've drawn 7, but that's because I ran out of space, so there's going to be 8 exons in the primary transcript of VEGF-A. So the portions that I've highlighted in red here, these are the exons. These are the portions which are actually going to translate, and the portions that have not been coloured in, which are between the exons, are the introns. And these are the junk DNA, effectively. These are the portions that aren't going to be translated, which don't code for a portion of a protein. Okay, so what's going to happen then is you're going to take the primary transcript and you're going to splice it, which is the process whereby you cut out the introns and then you stitch the exons back together. Now basically, there are alternative splicings of exon 6 and exon 7. These two particularly vary in how much of them you include. So basically, you can cut out different bits, well, differing amounts of exon 6 and exon 7. You don't have to include all of exon 6 and all of exon 7. You can cut little bits off, basically, and there are different ways that you can do that. And when you finally do produce a piece of mature mRNA from this primary transcript, which you get by sticking together the bits of the exons that you want to use, and I should state that most of the exons are essential, you'll need them, but for 6 and 7 you have many different splice variants. So you've now stuck your exons back together and you've just got a full piece that you're going to use for translation now. Okay, you can actually end up with lots of different mature mRNAs depending on how you actually splice the primary transcript. Okay, so it's this differing ways of splicing the primary transcript that then lead to the different isoforms of vascular endothelial growth factor A. So what are the different isoforms? Well, basically, even though I said well, actually, I don't think I have said it yet. There are 8 different isoforms. Oh, yes, I have said it up here. But 7 of them are actually going to result from different ways of splicing it. So let me write out the 7 that are going to result from different ways of splicing it. Okay, so they are called VEGF-A. So you always start with the VEGF-A, and then usually you subscript it with a number. So the first one is 121, and you might ask, well, what on earth does this mean? This relates to how many amino acids are in the VEGF form, basically, that we're talking about. So in this form, you have 121 amino acids. Okay, now what other numbers? I'm not going to write all their names out in full. Other numbers that you can have there, instead of 121, you could have 145, okay? You could have 148, you could have 165, 183, or 189, or 206. And all of these 7 different isoforms result purely from the fact that there are different ways of splicing, basically, the primary transcript, and therefore you can get 7 different mature mRNAs, which will make slightly different VEGF-As. Okay, and these are the 7 you can get from that. Now, you can then take VEGF-A 165 or VEGF-189, and you can chop them up, and they will produce an 8th type of VEGF-A. Okay, so both of them can be cleaved into the 8th isoform of VEGF-A, which is VEGF-A 110. Okay, so that then gives us 8 different isoforms of VEGF-A. So, these are the 8 different isoforms then of VEGF-A. Now, vascular endothelial growth factor A proteins do not function on their own. Instead, they assemble into dimers. So basically, you'll take one vascular endothelial growth factor A and you'll bind it to another vascular endothelial growth factor A, like so, to make a dimer. And you might be thinking, oh no, because there are 8 different isoforms and you might be able to mix and match them in the dimer, but thankfully, you don't. They are always homodimers, which means that the isoform that you have in slot 1 is the same as the isoform that you have in slot 2. So, for instance, you can make a dimer of VEGF-A 110, where you have a 110 in both positions, but you can't make heterodimers. You don't find VEGF-A 110 bound with VEGF-A 121. You find homodimers, basically. So VEGF-A, whichever isoform we're talking about, it doesn't function on its own. It always dimerizes with an identical protein to itself. And then you have this homodimer, and that homodimer is then going to function. So, basically, when we have a hypoxic cell, what will happen is that hypoxic cell will assemble hypoxia-inducible factor 1s in its nucleus, and these will increase the expression of the vascular endothelial growth factor A gene, and you'll get all of the isoforms of vascular endothelial growth factor A being produced, and they'll assemble into homodimers, and these will be secreted from the cell, and these are then going to diffuse over to the endothelial cells, and they're going to act on receptors on the surface of the endothelial cells. So we now want to talk about the receptors for vascular endothelial growth factor A. Okay, so, well, in fact, the receptors for vascular endothelial growth factor in general. The most important receptor for vascular endothelial growth factor A molecules is what's known as the vascular endothelial growth factor receptor, the VEGFR2. Now, there are 3 different receptors that are known as the vascular endothelial growth factor receptors. There is also a vascular endothelial growth factor receptor 1, VEGF1, and there is also vascular endothelial growth factor receptor 3. Okay, of these 3, vascular endothelial growth factor 2 is thought to be the most important in triggering angiogenesis, basically. There are also other receptors for vascular endothelial growth factor A molecules, and also for the wider family of vascular endothelial growth factors. They're not named vascular endothelial growth factor receptors, they're named other things, but they're still receptors for vascular endothelial growth factor. So examples, for instance, are the neuropilins. Okay, but we're not going to discuss those. We're going to concentrate on the vascular endothelial growth factor receptor 2, because this is the most important one in inducing angiogenesis or angiogenic results within the endothelial cells. So the endothelial cells will have vascular endothelial growth factor receptor 2 molecules on their cell surface membranes. So let's say this is a lipid bilayer of an endothelial cell, and let's show this receptor now in the lipid bilayer of the endothelial cell. So basically, its extracellular domain consists of 7 immunoglobulin-like domains. Okay, so let me put these here, and I'm not going to be able to fit them in. There's 6, okay, so let's add the 7th one up here. And then we'll have well, actually, that's it complete. So those are our 7 immunoglobulin domains. So these little balls here, these are what are known as immunoglobulin, or IG for short, and then like domains. Okay. Then we have our transmembrane region down here, which is the portion that straddles the membrane. That will be a membrane spanning alpha helix. Okay. Then in the cytoplasm, what we have is a split tyrosine kinase domain. We have these 2 portions of the tyrosine kinase which are split apart by a kinase insert, as it's called. Okay, so let's label these bits up, and then finally the carboxylic acid terminus of the polypeptide will be down here, so I should just label up that the amino terminus will be over here. Okay, so these 2 portions are the portions of the tyrosine kinase, so this is the split tyrosine kinase domain. Okay, so both of them are tyrosine kinase domains, basically. So this is the split tyrosine kinase domain, and then the portion that splits the 2 is known as the kinase insert domain. So this is the kinase insert domain. Okay, and then this other portion down here, which is after the second of the split tyrosine kinase domains, this is known as the C-terminal tail. Okay, and you might wonder, why am I telling you this? Well, because some of the tyrosine residues that are going to be phosphorylated on the cytoplasmic side of this vascular endothelial growth factor receptor 2 will be in the C-terminal tail. Okay, right. So there we have discussed the structure of a vascular endothelial growth factor receptor 2. In the next video, what we'll do is discuss how a VEGF-A homodimer is going to come and bind to 2 vascular endothelial growth factor receptor 2 proteins, dimerize them together, and then what will follow is autophosphorylation.